Hi, I'm Dave Sherwood from Test-All. I have a background of home inspections and environmental testing. What we're going to talk about in this segment is mold. As we get started in this, let me tell you a little bit about myself and where the background that I come from. After 35 years in doing home inspections, throughout that time period I've done research and gathered data in regards to mold situations that we have run into, testing, following up with remediation and results afterwards. I have taught for the EPA for the last 20 years in regards to standards for radon testing, mold, and lead. So I take that information, including other research that we've gathered from various sources, and put it together to create this class to present to you. We're going to start today with some of the basics, and we're going to go right to the more advanced stages and actual sampling techniques. My objective here is to be able to give you some specific tools that you can walk away with so that you can use in your company starting tomorrow. So let's start with the foundation, if you will, of this program, and that starts with the characteristics of mold. What is mold? So we're going to start with our first module, and that's a journey look into the world of mold. For that, we're going to talk about the different definitions of mold, because sometimes you get out in the field and somebody will communicate. In fact, the other day I was on a ride over to the facility here, and a gentleman said, well, mold, we work for this plastic company and we have molds throughout. They're not talking about fungal mold, they're talking about casting molds. So just understand that there are different definitions and you want to get on the same playing field before you get going and start talking about fungal growth. So those different definitions, as you'll see here, show that what we're really talking about is a very distinct furry growth and minute fungal hypha occurring typically in moist, warm conditions, especially on food and organic matter. Now, I will tell you that I was in a training class with somebody from the EPA who was reading this definition. So being curious as I am, I raised my hand and asked a question. The question was, so you say it has to be a warm, moist environment. So tell me, when I wrap food up and put it in my fridge and leave it for a couple of days, and then I take it out, I notice there's some furry growth on it. Are you telling me because it was in a refrigerator that that's not mold? He said, well, no, not exactly. I guess what I mean by warm is, say, above 40 degrees. Okay, so when I put food in my freezer and it's not wrapped properly or it's been in there too long and I get some furry growth on that, other than freezer burn, you're saying that that's all it is? He said, no, you can get mold on that too. Okay, so let's just say that it grows better in warm, moist environments. Okay, I can live with that. So let's understand that we want to make sure we understand where this information is coming from. And we want to be able to do and understand that this is a science. It's an ongoing, growing field. And our understanding of it is constantly growing and changing. So let's work with that and understand that the definitions are basic and there is room to grow within that. The next thing to take a look at is the CDC's description of fungi or fungi. And their definition is that it can be found both indoors and outdoors. We don't know how many species there are. It's tens of thousands to the best of our knowledge. Mold usually grows best in damp, warm environments and can spread by reproducing spores. Mold spores can survive harsh environments, and in fact, there's been a newer mold that they are working with, or a newer type that has been discovered indoors in buildings in areas like Phoenix, Arizona. One that has survived dry conditions. So those are things that we need to understand, and that there is an evolution process to mold. That, in other words, it doesn't just stay the same and it's always in the same environments. This is ever changing and evolving just as we are. So the question then oftentimes is, well, Dave, I got a lab report and it spells mold, M-O-U-L-D. They spelled it wrong, so I can't really trust the lab. It doesn't really work that way. You've got mold, which is your American and English version, M-O-L-D, but you've got a British terminology, spelled M-O-U-L-D, and they're both the same, mold or mold, tomato or tomato. And in fact, you have that with other things such as hyphae or hypha, and they're spelled a little bit different, same thing. So we look a little deeper into mold characteristics, and to do that, we're going to see what it takes for mold to grow. We're going to take a look at where do you find mold, what is the purpose for mold, and what is the physical makeup of mold. So in the characteristics of mold, mold is like a plant. Don't call it a plant because someone's going to call you on it, but it is an easy way to look at the components of it and relate it to something that we know and we understand. There are body parts to it that are similar to some plant life as we know it. So we'll do some comparisons there, but please don't call it a plant. It's a fungi. Next is mold grows, and mold can grow on almost any material. It oftentimes uses organic matter as its food source. Mold is not a particular color, and if you are using the term black mold, you need to understand that mold is not black. So we can throw out the thought process that if it's not black, it's not mold. In fact, I have seen mold in a rainbow of colors. I've seen it pink, blue, green, orange, brown, gray, white, and black, or at least what appears to be black to my eyes. If you actually blew it up, you'd probably see it was a dark purple, dark brown, dark green, and only looking black to our eyes, or because of the color of the lighting within the room that you're in. So you really have to be careful on using the terminology there. Next thing is you can have many different colors at the same time, at the same place. And oftentimes you could have the same species with different colors, or you could have different species with in the same location. Let's take a look at one of our molds, Penicillium. And Penicillium is one of the most common molds. And so you'll see in this slide here that it has a lot of the characteristics of this mold. We're not going to dive deep into these characteristics because for the purpose of identifying it out in the world as you're doing testing, these don't come into play, but they are certainly helpful and a big part of the science of it and for the lab people to understand that. What you'll notice, though, about something like Penicillium is it has its own characteristics, such as it grows very quickly. Typically, you can get mold Penicillium growing on a surface within 12 to 24 hours or 24 to 48 hours, depending on how much spores are in the air, how much moisture is there, and a suitable food source for it to work with. It is lightweight. The spores typically move about freely. You'll find it generally in a lot of different air samples. It needs less water to initiate its growth pattern than some of the other molds, such as Stachybotrys. So we take a look at this photo, and we see an orange here with some molds on it. When you look at that white mold, tell me what kind of mold you think it is. So it's Penicillium. But then it turns green. What color is that? And what kind of mold is that? That's Penicillium. But then you've got a different texture there. That different texture looks like it's a different mold, but it's actually still Penicillium. And this teaches us that you can get different colors, different textures with the same mold in the same or similar location. So don't try to identify it out in the field by how it looks. Here we have a slide of Penicillium lab picture, and then it shows it what it looks like in the real world. So there you've got one on that TV stand, but now you've got one in an attic rafter. That's also Penicillium. And so is this also Penicillium in an attic area. And so is this Penicillium. Notice how they all look a little bit different, but all of the tests show the same thing. Now, could there be other molds associated with it? Absolutely. But what I'm trying to point out here is that they can look very different and be in same or different locations and be the same type of mold. So don't try to identify that based on how it looks to you. All right, now we take a look at a picture, a slide here of Aspergillus. Aspergillus, as you take a look at it, looks very similar to that what you just saw with the Penicillium. In fact, oftentimes you'll see the labs showing Aspergillus penicillium being in the same identification. And the reason for that is because they look so similar. Their characteristics are pretty similar in its texture and in its way of being three-dimensional. But when we look at the characteristics for us out in the world and what makes it grow, there are some differences. Penicillium typically will take about 24 hours for it to start to culture. Not the same with Aspergillus. Aspergillus will take about 2 to 3 weeks for it to start its culture process. So what we're really starting to talk about is the difference between acute and chronic situations. And this is where it becomes important to us in the field. Acute being a quick snapshot event. One event that happened in a short period of time where you started to get mold to grow. So if you had an event where you had a water situation, things stayed wet for say 24 to 48 hours, you're more likely to get something like Penicillium growing in that type of environment. But then if you've got a longer chronic situation where moisture and say high humidity has been present for several weeks, you're more likely to get Aspergillus growing in that space. And if you carried it out even further and it was a little wetter over a longer period of time, then you're more likely to get wetter molds such as Stachybotrys growing in that environment. Now, could you have all three? Absolutely. And one could then go to another. Next characteristic I want you to take a look at is similar to a plant life. Mold has its cycles as it grows. So when you start out, if you've ever done a tomato plant or a potato plant, the idea is you take the seed or the potato and you put it in just water. It needs that like onslaught of water for it to start to grow the roots. Once it does, you transplant it into soil and have lots of water for that. And then as it goes on, a little less water over time is what it takes for it to continue to do its growth pattern. Mold can be very similar to that depending on the type of mold. It needs that onslaught of water or it needs a lot of moisture over a long period of time for it to start its growth pattern. But as it starts to mature, it needs less moisture than it did to initiate. So just some identification patterns similar between plant life and mold or funguses. The next one is, what does it take for plants and funguses to grow? Well, no different for plants and funguses is human and animals. Meaning, what does it take for us to grow? It takes food, it takes air and moisture or water source. So because of that, when you say, well, what does it take for this fungus to be here, for this mold to be present? Remember that it is very much like humans. It takes your air supply, your food supply, and your water supply for it to continue its growth process. When we look at the food supply for molds, it will grow on almost any organic or feed off almost any organic material, such as paper, cardboard, wood, wallpaper, glue, and even dust. And so when we start thinking about dust landing on a surface area, it really starts to show that you can get lots of food supplies almost anywhere. And as clean as somebody can be, if you've got enough dust in the air that lands on the surface area and you've got the moisture that it needs, and we've got air everywhere, it's possible to get the mold culturing and growing in that area. So we really look at how do we control this mold situation. Well, air is air. Air is everywhere. So we can't really control the air. We can't really control the food supply as well as we would like. So then that leaves us with water. Water is the most controllable portion when we talk about the control of mold or the prevention of mold growth. When people look at their homes and say, I'm really concerned about mold being in their home, they're not really concerned about spores moving about freely in the home because they're always there. What they're really talking about is mold growth in their home. And so that's really what we as inspectors and testers are looking for, is the potential for mold growth within the home. When we look at water and we look at how do we control this mold growth, what we're talking about is water flow, water absorption, water condensation, and then water humidity. These are all the various ways that you can get moisture introduced to an area that could initiate or maintain mold growth. When we look at an annual cycle of a plant such as this one, you're going to see the plant on one side is dry and colored brown, and right next to it is it in the springtime with it turning color. Molds will go through the same type of cycle, not seasonal changes as we know it, but changes where it can change color as well. So you have green and then purple here, or you might have brown and green. And so there are stages that funguses go through where as it's growing or culturing or changing in its cycle, it will change colors. So it will look different to us, including its flowering stage or fruiting body type stage, and then of course its sporing stage. And all of those can make a difference in how it looks to us and how it matures in its cycle. Okay, so then we look at the purpose. We recognize that mold has a purpose. It's in our environment everywhere we go. And people say, well, I want to get rid of it, or I don't want any mold in my home. And when you have a client that says that, we have to understand and remember what we're really talking about and help educate them. Mold really takes on a purpose in our world. This purpose is to help break down organic matter. We're looking at the role of fungi such as molds, yeast, slimes, mushrooms. In the cycle of life, it really decomposes organic matter, breaks it down to carbon, and that acts as a recycling agent, so to speak, for our natural environment. So in the outside world, it's doing just what it's supposed to do. It's been around for a very long time and will continue to be around for a long time, just as you walk through the woods and you have all the debris on the floor of the woods. If that debris was not being broken down by something like funguses and molds and such, we would have debris building up all of the time. We wouldn't be able to walk anymore. Can you imagine what our planet would be like if we couldn't decompose things like fruits and vegetables and leaves and sticks and grasses? We'd really have a whole mess on our hands. And so we want to befriend this mold and this fungus, but in the outside world, not in our safe haven, not in our homes and buildings where we want to work and play and reside. That's when we look at molds and funguses in our world, and one of the most misunderstood definitions is a mold and a mildew. And so here we show that you've got a living plant that's got a fungus on it, and you will find mildews growing on living plants. When we look at decomposing plants and tree parts and such, we'll see these molds and funguses growing on them. And so that's what we're really looking at, is the organic matter that we have where the mold is feeding off it, and looking at those differences. When we look at fruits and them decomposing, we can see the different molds growing on it to help decompose that fruit. So let's take a look at the parts of mold, because this is an important aspect. Now, what's really important about this part of it is that you're going to see that different mold labs will have different names, and that's okay. We're going to talk now about this particular lab that comes out and identifies that stolon is the interconnecting pieces, and the spores are the dried fruiting parts, or the seedlings as we know them in plants, that start to move about freely and spread so that it can continue to grow in culture. Then we go to the next one, and it shows a little bit of difference in its body parts, but they're the same. I don't want us to get too hung up on those names. The names that I really want you to understand for this training purposes are the two main body parts of hypha or hyphae and spores. The spores are the fruiting bodies that have converted to seedlings and then move about to, again, reculture the molds so that it can continue to grow.